second major biomolecule that we will discuss will be on proteins are among those four major biomolecules, proteins have the most diverse in function. Okay? So meaning daghan kayo sila og function. So proteins are the most structurally and functionally diverse group of biomolecules. Okay, so for function, they are involved in almost everything like metabolism, support, transport, regulation, and motion. Okay, so they function as enzymes, or they're called biological catalysts. And these biological catalysts speed up chemical reactions. Okay, so for example, sa atong na-experience na, we have digestive enzymes. Those enzymes are actually proteins. So like amylase, diba? We've had this in carbohydrates. We have the salivary amylase. So you have amylase, you have lipase. This is the enzyme that breaks down lipids. So lipids, lipase. We have lactase, we have protease. Okay, so in molecular biology, we also have polymerase. So if in the COVID-19 RT-PCR, in RT-PCR for detecting the SARS-CoV-2, you need to use DNA polymerase and RNA polymerase. So those are actually proteins. So enzymes are proteins. Okay, we also have ligase, and then in the industry, we have dairy, baby food, rubber, beer, photography, contact lens cleaner. So those things are actually those things involves the use of proteins, enzymes as biological catalysts. Okay. So this is how they. Let's just have an overview because we will have a separate discussion on enzymes later. So let's just have an overview. So this is the substrate or your reactant. Ang substance na actant sa enzyme. So this is the enzyme. The gray one is the enzyme. And you have the substrate. So this space here is called the active site of the enzyme. So ang active site class, it's the site nga directly nga mo interact with the substrate. So when the substrate and the active site of the enzyme binds, so the substrate does not just bind to any part of the enzyme, it binds to the active site of the enzyme. Okay, so when that binds, it causes the lowering of the activation energy. So it actually makes this substance more reactive. So that's why the reaction goes faster. So manang mo speed up siya og chemical reactions. So after that, it could form the products, and then the enzyme will leave. The products will leave the active site of the enzyme. So that's how enzymes work as biological catalysts. Okay. So proteins also work as for support. So we have structural proteins like in keratin. Keratin is a protein present in hair and nails. We have collagen. Collagen. It supports ligaments, tendons, and skin. If you read kanang mga advertisements like or labels sa kanang like mga lotion, a lot of lotion says that it has collagen. So it's actually support. It's actually a protein that acts as support for the skin. And then we have silk. It's still protein present in cocoons and spider webs. Okay, so Monel sa collagen collagen filler treatment. So before and then after, her lips were and her skin were very different. Look at her skin. You have the hair and the spider webs. Okay, proteins also acts for transport. Okay, so in the cell membrane, so sa cell membrane, you have this lipid bilayer. So these are lipids. We haven't discussed about lipids, but we'll discuss them after proteins. So we have this lipid bilayer, and then not all, diba, this is the cell membrane. So not everything could pass through the cell membrane. So a lot of things actually needs proteins. So these proteins acts as transport proteins, which means sila ang mupasulod o mupagawas sa substances nga dili makapass through ani sa lipid bilayer. Okay, so these are channel and carrier proteins in the cell membrane. So ang function na nila is to allow substances to enter and exit the cell, because many substances are too small or too big to enter the cell, diba? Of course, if it's too big, it could not possibly pass through this lipid bilayer, diba? So they need transport proteins. So we also have transport molecules in blood like hemoglobin. This transports oxygen in the blood. We know that, right? So this is the structure of hemoglobin. Okay, you have two alpha chain, this violet and this orange one, and then you have beta, this red and the green one. Okay. Proteins also acts as for defense. So it's present in antibodies. So the function of those antibodies, as you know, is to combat bacteria and viruses, diba? So these are the ang sa structure sa antibody, no, you know. So we have, these are variable regions, kining red. So constant ni ang kining blue. This is constant region. So ang mag-change ani is kining red. So it could produce this one generation of oxidants. It also acts for immunomodulation. You have opsonization, activation of complement, virus and toxin neutralization. So depending on unsa ang gibutang diri sa kaning variable nga region, it could make different functions. So antibody dependent cell cytotoxicity, direct antimicrobial activity, you have reduced damage to host inflammatory response. So it's actually one of the function of proteins. Okay. Then we have for regulation. So most important are hormones and receptor proteins. So these hormones are actually proteins that are used for regulation nga process processes. So we have intercellular messengers that influence metabolism. Like we also have insulin, as you know. These are proteins that regulates the amount of glucose in the blood and in cells. We've encountered that when we discussed carbohydrates. Then we have the human growth hormone. So its presence determines the height of an individual. That's what we call the growth hormone. Okay. And then we have receptor proteins. From the word itself, they are proteins. So they, from receptor, meaning sila ang murag mo detect. Okay. So built into the membranes of nerve cells. So na sila sa membrane. So they will murag sila ang mo kana sa stimulus gani. Stimulus and response. So sila ang these proteins are the ones that detect the chemical signals. So that's why they're called neurotransmitters. And or the neurotransmitters, that are released by other nerve cells. So from one nerve cell to another nerve cell, receptor proteins are needed para matransmit ang unsa nga chemical signal that is released by one nerve cell para ma-pass on siya sa other nerve cell. Diba? Okay, so wait, this figure class in a picture shows several organs that gives off proteins. So we have adrenal, this gives off hormones, right? So pancreas, as you know, insulin is produced there. You have thymus, parathyroid, thyroid. So these organs produces proteins, especially hormones. Okay. And it's also involved in motion. So, for example, a muscle contraction, it uses up actin and myosin. These make up muscle fibers. So these are proteins also. So imagine the diversity of the function of protein. We have motor proteins within the cell. So this is within the cell. And these motor proteins allow the cell components to move from place to place. So for example, you have flagella. It moves, if you read about flagella, it, mauna siya ang mag-cause para move ang cell. Okay, so we have cilia, and this moves the contents around the cell. Okay, so now that we know about the function of different functions of proteins, let's have the structure. So the structure of the proteins determine its function. Okay, so it's very dependent with the structure. Okay, the function of the protein. It's very highly dependent on the structure of the protein. For proteins, you have a monomer. Ang monomers of proteins, because these are polymer, the proteins is a polymeric molecule. So the monomer is there are the amino acids. These are the building blocks of proteins. So we have 20 different amino acids. Ang 12 ana are made by the body, and ang 8 ana are called essential amino acids. They are called essential because they need to be taken in from the diet. So they must be taken in from food. Okay, that's why they're called essential. Okay, so from monomer nga amino acids, you can form them into a polymer, which we call as a polypeptide. So this is how it works. We have one amino acid and a second amino acid, and then it releases water, as you know, formation of forming a bigger molecule. You need to dehydrate. Okay, so you need to release water, and then you form the polypeptide chain. So this is already a polypeptide. Specifically, this is a dipeptide. Okay, so protein can be one or more polypeptide chains folded and bundled. Okay, like for example, growth hormones. So imagine the complexity of the structure of a protein. So in any structures of growth hormone. So these all of these are peptide bonds. Amino acids bonded together by peptide bonds. Another example, hemoglobin. So imagine katong duha ka alpha, so you have an alpha, beta, 2 alpha and 2 beta subunits of hemoglobin. So these are all proteins. So proteins are large and complex molecules, which is why we have we try to deduce this structure, this complex structure, by using different levels like primary level, secondary, tertiary, and quaternary level of structure. Kay para mahinay-hinay na to og deduce ang complex structures of protein. Okay? So it is a complex 3D shapes. This is another protein. This rubisco is another example of protein. So amino acids have the structure. They have the central carbon, which is called as the alpha. This carbon is called the alpha carbon, and then we have an amino attached to that alpha carbon. Okay, so this is amino and the alpha carbon, and then we have a carboxyl group. So this is the carboxyl group. So this is the structure of amino acid. Okay, so we have this alpha carbon, this amino group, and we have the carboxyl group, and lastly we have the R group, or the side. So this is the side chain. The side chain is what differs each amino acid from each other. So remember that we have 20 amino acids. So this one is maintained, but then each amino acid will have different group. Okay, so this is the variable group. So this because this is the only thing that's variable in each amino acid. This R group confers the unique chemical properties of the amino acid. So this is what distinguishes one amino acid from another. So that's why in most books they actually classify amino acids. So we classify them as non-polar amino acids. So glycine is non-polar. So the R group, this is highlighted is in white. So muna ang R groups. Okay, so ang glycine, you only have H. That's why it's non-polar. You have alanine, valine, leucine. Look at the R groups of these non-polar. Isoleucine, so they're the same, huh? And then methionine, you have phenylalanine, you have tryptophan, and you have proline. Imagine, I mean, if you think about it, these are basically just hydrocarbons, right? Except for this one, of course. But still, this is non-polar because as these three the S here does not really contribute a polar bond because it's just bonded to CH2 and CH3. Then we also have polar and charged. So for polar, we have CH2OH. As you know, this makes it polar. You have threonine, so the OH makes it polar. And then CH2, this SH makes it polar. Then we have tyrosine, this OH makes it polar. This one, the amide, makes it polar. And this one, another amide makes it polar. Then we have electrically charged. So electrically charged, these are aspartic acid. This aspartic acid makes it polar. This carboxyl group in the aspartic. And then we have glutamic. These are acidic, okay? And then so electrically charged can either be acidic or basic. So we have lysine. So lysine is a basic amino acid. So we have NH3 plus here. You also have this amide here. Sorry, it's not an amide. It's an arginine. It's just a an amine. Yeah, it's just an amine. And then we have histidine. So they're polar because of these charges. Okay, so you have positive, positive, we have negative, negative. Okay. We have, as you notice, we have two sulfur-containing amino acids. We have methionine, which is non-polar, and we have cysteine, which is polar. Diba? So these could form disulfide bridges. So this SH here, could bond with another. So mo dehydrate na. Ma-remove na ang H dira, and then it could form another S. Okay? So, dili ma-remove ang H. Mo-form na siya og another SH. That's why it's called disulfide bridge. Okay? So, duha ka S, like this one. And we also have methionine. Okay? So, it forms cross links between sulfurs in amino acids. So, this is a very important nga interaction class because this gives a variation in the function of a protein, manggod. So remember that structure determines the function, okay? So that's why if you wonder why perms smelled like rotten eggs, diba? Ang anak mo sa salon, then ay nagpa-perm, it's actually because of this disulfide linkage present in cysteine. Okay, so ang kanang nasa perm class, it contains the as you know, it contains the proteins, and keratin contains this sulfur containing side chains. That's why ang perm it smells like rotten eggs because sulfur smells like rotten eggs. Next, so How do we build proteins? Gikan sa amino acids, diba? So the bond formed is called peptide bonds. Okay, so this is the linking of amino of one acid, the amino group of one acid, to the carboxyl group of another. So for example, as what I've shown you earlier, kine. So we have one amino acid. This is amino acid 1 and this is amino acid 2. So okay, alpha carbon. Remember, alpha carbon, carboxyl group, amino group. Alpha carbon, carboxyl group, amino group. So, ang carboxyl group o ang amino group, you take water, dehydrate it, and then there you will form the C-N bond. So, you have C, single bond, N. So, this is what you call now as the peptide bond. Okay, so it's this one. The same as this one. So it forms a peptide bond. So ang peptide bond class is the C double bond O NH kana nga bond. This is the peptide bond. Okay? So what if sa carbohydrates you have a glycosidic bond or a glycosidic linkage, in proteins it's called the peptide bond. Okay? So ang peptide bond, if you notice, it's actually just an amide bond. Diba? C double bond O in NH. It's called an amide bond. Okay, so that's why it's a C-N bond. And you read this amino acid. This amino acid is written from N terminus to C terminus, which means ang amino group dapat ang nasa left side and carboxyl group ang nasa right side. Alright, that's how an amino acid, a polypeptide is written. So this is what you call as a dehydration synthesis because you are removing water. Okay, so here you have glycine. So this is glycine and you have alanine. So you remove water. So you form this peptide bond. So because this is from glycine and this is from alanine. And you read this from N to C terminus. So this is the C, di ba? Kaya nade ang negative, nade ang positive. So dapat you need to read this from N to C. That's the nomenclature of polypeptide, okay? So in naming this polypeptide, you name this as glycyl alanine. Okay, so ang katong mga gauna, so except sa last one, you change the name to YL. You remove the INE, change to YL. So glycyl alanine. Okay? So valyl glycyl alanine without any space. Okay? So that's how you name a polypeptide. So kung napanay valine dira gauna, you have valyl glycyl alanine. Okay? So that's how you name a polypeptide. So for example, here you have a in this one, it's called a dipeptide because you only have two. You only have two amino acids. Okay, you only have glycine and alanine bonded together. If you have four, it's called a it's already called a polypeptide. Okay, so it's not a dipeptide or tripeptide. It could be it's a tetrapeptide, but it's you can already classify this as a polypeptide. Okay. So every peptide bond, you release water. Okay, so you release water for every peptide bond formed. So and then when you write these amino acids, these amino acids are formed from N terminus. Remember, N terminus of the first amino acid and the C terminus of the last amino acid. And then the R groups will be written the amino acid R groups, so the side groups, will protrude in an alternating manner. So if you notice, ang R group na diri, R group, R group, R group. This is a very convenient way to draw structures of polypeptides. Okay, so you have N, so you have amino group alpha carbonyl, amino group alpha carbonyl, amino group alpha carbonyl, amino group alpha carbonyl. Okay, so it's very convenient, diba? it's very convenient to draw these amino acids. So remember, you need to the R groups protrude in an alternating manner, and then you read this from N terminus. You also name this from N terminus to C terminus. Don't forget that, ha. N terminus meaning na ang amino acid na adere nya carboxyl ang nadere sa last. Alright? So these are the non-covalent bonds within and between V change, polypeptide chains. And these are as important in their overall conformation and function. Okay, so aside from covalent bonds, peptide bond is a covalent bond, diba? It's a sharing of electron. So aside from covalent bonds, we also have ionic bonds, hydrogen bonds, and van der Waals forces. Okay, so ang ionic bond, so from the side chains, for example, from the side chains, from the side chains of amino acids. So you have, for example, for carboxyl, the acidic amino acids, diba, you have this negatively charged, and for basic, you have positively charged. So they could actually form ionic bonds. Okay? So that's why if naani silang duha, if naani nga side chain, if they meet each other, okay, so that's why you form this, mo form siya og U kay because of this attraction, this ionic attraction here allows the formation of this U nga conformation. So mao nang if you notice the structure of protein, it's ana-ana, di ba? It's very complicated. So you also have hydrogen bonding. So these are hydrogen bonding. You have van der Waals interactions. Okay? So you can have hydrogen bonding from side chains. So these are side chain hydrogen bonding. And you can also have hydrogen bonding nga gikan sa imong carboxyl o gikan sad sa imong amino. Katong sa main nimo nga polypeptide, diba? You have a carboxyl group and an amino group. So you can have this hydrogen bonding, or you could have this side chain nga hydrogen bonding. So you have side chains nga maka-form og hydrogen bond, diba? So you can also have hydrogen bond nga gikan jud sa main nga carboxyl ug sa amino. Okay? So the side groups of the linear unfolded polypeptide are intermingled. Only when correctly folded do we see the wonder of nature. Okay, so they're not really kanang in ang kaning mga side chain class. These are the side chains, okay? Kaning blue, green, okay? So these are not actually in kanang straight nga form, okay? This is the unfolded nga polypeptide. So in nature, actually, when they are correctly folded, so mao na ang kanang functional nga structure of this polypeptide. So you have hydrophobic. So ani sa sulod magtapok na ang hydrophobic. So katong mga non-polar nga side chains. So you have non-polar amino acids, diba? So ang katong mga non-polar, so mag-meet na sila sa sulod, and then ana sa gawas ang mga polar nga side chains. And then they can form hydrogen bonds, they can form the katong disulfide bridges, ionic bonds. Okay, that's why, because of those different interactions, that's why ang structure sa protein is this complicated. Okay? The locations of the hydrogen bonds are not restricted to those between the side groups. Okay, so hydrogen bond between atoms of two peptide bonds. So as I've said, dili ra sa side groups ang hydrogen bonds. Okay, it's not just for side groups. As I've said earlier, naapoy hydrogen bond nga gikan jud siya sa amino acid nga portion ug sa carboxyl nga portion. So hydrogen bond between atoms of two peptide bonds. So gikan sa peptide bond jud siya, ha. Sa peptide bond. Hydrogen bond between atoms of a peptide bond and an amino side chain. So gikan sa peptide bond, so if you notice sa side chain nya gikan sa peptide bond. So kini gikan ni sa peptide bond nya kini sa side chain. So they form hydrogen bond. Okay, so kini sa side chain ni puros, sa sa main jud amino acid o carboxyl both, okay, or amino both. Okay, so you can form a hydrogen bond for that. And then you also have hydrogen bond between two amino acid side chains. So for this one, you have just all of these are side chains. Okay? So puro side chain nga amino nga peptide bonds. Okay? This blue one. So imagine kining mga blue, yellow, o red, pink. These are hydrogen bonding nga different different components. So you have hydrogen bonding nga gikan sa peptide bonds, you have hydrogen bonding nga gikan ra sa side chains, and then you have hydrogen bonding nga gikan sa peptide nya ang iyang ka hydrogen bond is gikan sa side chain. Okay? So these hydrogen bonds actually forms the secondary and the tertiary structure of a protein. So next, the 3D folding of a polypeptide is governed solely by the sequence of the amino acid. Okay, so this folding is governed by the sequence of amino acid. So under some physiological conditions and in vitro, so many polypeptides can reversibly unfold and refold. So you have, this is the like the natural or the this is the kanang active nga form sa protein. So this is the 3D nga conformation of that protein. So this is the protein isolated from cells. And then when you expose this to high concentration of urea, you can do this in the lab. If you expose this to high concentration of urea, you actually denature that protein. So denaturing a protein, it means nga matangtang iyang 3D structure. You can form a linear na nga protein. And then if you remove urea, you can form that back kay mobalik raman pud tog interact ang mga whatever is interacting in there, like the H bonding, ionic bonding, the disulfide. Mobalik raman pud sila, so that's why mo reform ra ang protein. Okay? So proteins are actually very interesting biomolecules. So when these proteins class are already these amino acids are already formed as polypeptides. So after sila amino acids, diba, amino acid will form peptide with another amino acid. So when these amino acids are already incorporated as polypeptides, naiuban na nang will be modified. That's what we call as post-translational modification. So meaning after translation. So ang different post-translational modifications nga ma-experience nila is, for example, addition of phosphate groups. You have addition of methyl groups, addition of hydroxyl groups, and formation of disulfide bonds. Okay, so these post-translational modifications are happen na naka-form na og kanang polypeptide ang mga amino acids. Okay, so some of them will be attached with phosphate, attached with methyl or hydroxyl. Okay, so they're not really just katong 20 ka amino acids. Some of them, so some of the proteins are actually modified. So the process is called the post-translational modification.